All right, assuming you're comfortable with mitosis, we're now ready to go ahead and proceed to meiosis. Now, meiosis, good news is that it's not really all that much more difficult than mitosis, but there are a few extra steps and there's a few extra things we have to be careful about. Now remember, the purpose of mitosis was to go ahead and make replacement cells. The daughter cells were supposed to be completely and totally genetically identical to the parent and to each other. Here, we're specifically looking for gametes, or sex cells. Now, these things have to be different in that they have only one set of chromosomes. In this case, we call those haploid cells. Now, for our purposes, the only haploid cells we're going to deal with are those that are going to be destined to be gametes. Normal somatic cells, cells that make up every other part of your body, are diploid, two sets of chromosomes, one from each parent. And of course, we all start as a diploid zygote. All right, so right off the top, we're going to have to worry about something just a little bit different. In mitosis, we didn't make any distinction about the types of chromosomes you were looking at. We were just worrying about the total number. For instance, humans have 46 chromosomes. But, hey, 23 of those chromosomes originally came from your mother, and 23 of those chromosomes originally come from Dad to give us those 46. So it turns out that we can pair up these chromosomes with these chromosomes and end up with two sets. Now, the pairs that we make are alike in size and shape and in the amount and type of genetic information that they carry. If you're diploid, that means you have two of these homologous pairs. If you're haploid, or if the cell is haploid, you only have one member of each pair. So let's take a look at these chromosomes. On the right, you've got a karyotype, or a picture of what human chromosomes would look like if we just kind of arranged them in sets, or as homologs. Now note that they're coming in pairs, 23 pairs. We have one little mismatched pair over here, and that's due to the fact that this individual is a male. Females will have 23 pairs of matched chromosomes, males 22 pairs, and then one mismatched pair. More about that in a later unit. But if you look at each individual chromosome, just one chromosome, over on the left-hand side, you can see that, just as we said before, you've got two chromatids joined by a centromere. But maybe this chromosome with the arrow in red came from your father. This chromosome with the blue arrow, or with the green arrow, comes from your mother. You might have a gene over here which codes for eye color. Well, you have a gene over there that codes for eye color. One gene from your mom, one gene from your dad. You might have a gene down here which codes for, oh, I don't know, length of your small intestine. And guess what? There'd be another gene over there that would also code for length of your small intestine. Two bits of genetic information, two sets of genetic information, one from each parent. Now, in meiosis, you're going to have two separate divisions, and both of them mimic the events of mitosis, but they're not exactly alike. So we're going to refer to them as the first division, meiosis 1, and the second one, meiosis 2. Now, the first division goes through stages very similar to interphase and prophase of mitosis. You still have G1S, G2 of interphase. You still have replication of the genetic material. You still have the chromosomes existing as two chromatids joined by a centromere. But the big deal. Here in prophase, very early prophase, the chromosomes will begin to group together, and when they appear in prophase, they'll come together as homologous pairs. So the cell down below is a little bit simpler than us. It just has 6 chromosomes, and its diploid number. But note that we've got 3 pairs of homologous chromosomes, and they're arranged as pairs, side by side. Since each chromosome has 2 chromatids, that whole structure would be a tetrad. All right, another very, very important thing happens in prophase. Not only do the chromosomes line up side by side, paired up as homologous pairs, but oftentimes they lie so close to each other that they will go ahead and overlap, kind of stick to one another. Note the large blue chromosome on the left-hand side over here. The right chromatid and the left chromatid have the same gene sequences, denoted by a capital A and a capital B. It's duplicated on the right-hand side and the left-hand side. The green chromosomes, now, lowercase a, lowercase b, and duplicated lowercase a, lowercase b on the other side. When these things lie side by side, touching each other, sometimes bits and pieces of one chromosome actually rip off and exchange places with bits and pieces of another chromosome. The result, or on the right-hand side, over here, is that on the right blue chromatid, we now have a big A gene attached to a little B gene. It wasn't like that in the original chromatid. Note that the left and right blue chromatids no longer resemble each other. They're no longer the exact same duplicates. Same way with the green chromosomes. Right? This is something called crossing over. And it sounds bad, but this is supposed to happen. This is designed to happen. As a matter of fact, it's a good thing that it happens. Crossovers contribute to genetic diversity. And hey, the more crossovers that occur, the better. Bottom line, at the end of all these crossovers, which occur completely and totally randomly, you're going to end up with a guarantee that all of your chromatids are going to be different or carrying different genetic information. Don't even worry about this slide. Dude, cross it out. It doesn't exist. It's a figment of your warped and fevered imagination. Shoot, I wish I had erased this beforehand. Oh well. Okay, so after prophase, we're supposed to have metaphase. Well, same thing in meiosis. After prophase 1, you got metaphase 1. The chromosomes are going to line up along the equatorial center of the cell. But here's the big difference. They're lining up, still paired up as homologs. Also, at the end of metaphase, beginning of anaphase, the centromeres aren't going to replicate. This is going to really, really be important later on. Notice the orientation that we have here, right? The cell has its chromosomes lined up so that the chromosomes are lined up 2 by 2. If we had the same thing in mitosis, right, the chromosomes would be lined up so that they would essentially be single file, with the spindles going out in this direction. Right. This is going to make a big difference in what happens next. All right, now in anaphase, since the centromeres do not replicate, that means the chromatids cannot separate. They have to stay together. That which moves to one end of the cell or another is the entire chromosome, each with its own centromere. So this group of chromosomes and this group of chromosomes are going to end up in different cells. All right? Now note that you're going to have, well, you start out with 6 chromosomes. Over here in cell A, you're going to end up with 3 chromosomes. In cell B, 3 chromosomes. You're already haploid because you have half as many chromosomes, but each chromosome still consists of 2 chromatids joined by a centromere. So, yeah, you're going to have to go through a second set of division. Right? We'll go immediately from a telophase into, a brief, sometimes they call it interkinesis. You don't really have to go through anything like G1, G2, or S. Hey, everything's already been duplicated. We just need to go ahead and redistribute our genetic information. All right, so I switched colors on you here, but let's go ahead and review what we got so far. So, different view of the same cell. Still had 6 chromosomes. We're going to go ahead in prophase, line up, group together as homologous pairs. You might have some crossovers going on. If they occur, they're going to occur here. In metaphase, the homologous pairs of chromosomes are going to line up 2 by 2. And then we're ready for anaphase. Once again, in anaphase, since the centromeres don't replicate, the chromatids can't separate. And what migrates to each pole, up at the top and bottom of the cell here, are going to be the 2 chromatids joined by a centromere. Now, the resulting cells, after telophase 1, are going to have 3 chromosomes, 1 member of each pair. Technically, they're going to be haploid, but each chromosome still contains 2 sets of genetic information. So we're going to have to go through a second set of divisions to go ahead and take care of this. This is what goes on in meiosis II. So a quickie overview here. Everything we see on the left half here is stuff that went on in meiosis I. But we're going to go ahead and get rid of our extra set of chromosomes here, or extra set of chromatids, I should say, by doing a second set of division. So each cell is going to go ahead, go through what looks like mitosis, because essentially it's the same steps of mitosis, minus interphase. Right? The cells go into prophase 2. In prophase 2, hey, you don't have to go ahead and line up as homologous pairs because there are no homologous pairs left. You've only got 1 member of each set of chromosomes. When we go ahead and do metaphase 2, hey, the chromosomes can't pair up, so they just line up single file just like they would in mitosis. In anaphase 2, well, the centromeres can replicate, the chromatids do separate, so what we end up with in telophase, or telophase 2, is, hey, each cell having half the number of chromosomes, and because of the way the chromosomes were slightly reshuffled in prophase 1 with crossovers, each of these cells will be genetically unique, genetically distinct. All right, this is just a quick review of differences, basic differences between mitosis and meiosis. I'll let you read it for yourself. But the one thing that we want to go ahead and hammer here is that because of the crossovers that occur here but not here, endpoint of meiosis is going to give you completely and totally genetically distinct and unique cells. Not so in mitosis. In mitosis, they should be just carbon copies. Now, I did leave one thing out, and when I get back to class, I'll show you what happens in metaphase 1, which really contributes to genetic diversity as well. And between the two of them, you're almost guaranteed that all sperm cells and all egg cells, even from the same person, are going to be completely and totally different. All right, one last little bit. Turns out there's going to be a little bit of difference, at least in mammals, in whether or not you're making sperm cells or egg cells. So spermatogenesis, formation of sperm cells, right? Obviously, this is going to happen in the testes. Testes have, each testis has got like 1 million of these coiled seminiferous tubules where you're going to have not only hormones produced but sperm. And your sperm are produced pretty much continuously from puberty till death, as long as you've got decent levels of testosterone. Now, peak sperm production is probably in your, in your 20s. And it starts to go downhill from there, but essentially you're creating sperm forever. Anyway, within the testes, there will be diploid cells called spermatocytes. And we'll start out with the primary spermatocyte, diploid. It's going to go through the first meiotic division, meiosis 1. Then you'll get secondary spermatocytes. And you'll immediately go into a meiosis 2, where you'll end up with spermatids. Spermatids are immature sperm, and they're going to be stored temporarily in the epididymis. This is some coiled structures just at the base of each testis, or at the top of each testis. And eventually these things are going to mature into all the structures that they need to have functional sperm cells. So in male, it's straightforward. Diploid primary spermatocyte, meiosis 1, secondary spermatocytes, meiosis 2, 4 spermatids. All right, the female counterpart to spermatogenesis, oogenesis, of course, happens in the ovaries, but this happens during the development of the female before she is born. In other words, you're busy making eggs prior to your own birth. So it starts out similar. You'll start out with the primary oocyte, and primary oocyte will go through meiosis 1, but we'll have an unequal division of the cytoplasm. This is almost a little bit like budding. And most of the cytoplasm is going to end up in a secondary oocyte, and what's left over gets into this little polar body. And the polar body isn't necessarily going to be used. It's not going to develop into a mature egg. Now, here, meiosis kind of sort of stops for a while, and can stop until just prior to ovulation. Just prior to ovulation, then the cell is going to divide again. Now, a polar body might divide, but all you're going to get is more polar bodies. Don't need them. The secondary oocyte will divide unequally, and again, you got a polar body, and an ootid, which is a immature egg. This will eventually mature and be released in ovulation. So more about this in class, but I want to give you guys at least the basics so that you've got some of this stuff down before we head on to bigger and better things.